In this video, we're going to talk about sex determination, but first I want to define a few things. There is a lot of talk in the media about sex and gender, so let's just describe those things first. First off, when we talk about sex, we're talking about biological sex, which are the classifications of male and female, and this is based off of chromosomes and anatomy. Then there is a second definition here, gender. Gender is a social construct. Gender is defined by the people around us, our society. It's based off of many things, including biological sex, including gender roles that society has constructed, and also including self-identity. And so this is a biology class. We're going to focus on the biological sex part. Sex can be based off of chromosomal sex. For a female, we have XX, and for a male, we have XY. However, as you may have gleaned from our last karyotypes video, you could possibly have triple X or XXY or XYY or XXXY or any other combination here. And so it's not always just black and white where there is just XX and XY. Similarly, we can define sex from anatomy. And so we base it off of what genitalia do you have? Do you have male genitalia or do you have female genitalia? But in this video today, we're going to talk about how there are actually areas in between, which we consider intersex. And so what you're going to learn from this video today is that there is nothing that is so black and white, so binary that we have known. There's actually a lot of in between, which we will discuss today. You're welcome to look through the slides about gender. This is not, an anthropology or sociology class, so we're just going to focus on the biology part, but it is interesting, so you're welcome to review those. So what we're going to focus on is how is sex determined? How do we determine sex? There's two possible causes for sex determination, whether we decide someone is male or someone is female. And so we know a male would typically have XY, a female typically has XX. And so that means we can define a sex based off of two possible causes. The presence of a Y chromosome, and if they have a Y chromosome, that may mean that they're male, or the number of X chromosomes, and that may mean if they have two X chromosomes, that means they're female. So if the Y chromosome determines sex, that means Y always means male. No Y always means female. So somebody who is lacking a sex chromosome and has a karyotype of 45 chromosomes and only one X, that individual should be female. Somebody who has an extra sex chromosome, so 47 chromosomes with an extra X, but XXY, that person should be male. If the number of X chromosomes determines sex, that means that two X's means female. That means one X, because this male only has one X. That means one X means male. So that means that somebody who has 45 chromosomes and only one X should be male. And that means that someone with 47 chromosomes and an XXY should be female. Well, from what we have seen in our last karyotype video and karyotype slides, somebody who has 45 chromosomes, and so they only have one X chromosome, they are female. They are phenotypically female. They have the anatomy of a female. This person has Turner syndrome, and they may have some developmental changes, but they are female. And so what that means here, what that supports, is that a lack of a Y chromosome means a female. We also see that somebody who has an XXY, so they have an extra X chromosome here, they are not phenotypically female. They are phenotypically male. They have male anatomical parts. And so what this tells us, these two bits of information tell us, is that the presence of the Y chromosome dictates the sex. And it dictates whether someone is going to be male or female. That means that the other possibility, the number of the X chromosomes, does not tell us sex. The Y chromosome is going to tell us sex, and it tells us if somebody is a male. So, our Y chromosome is going to tell us the sex of an individual. If you have it, you are phenotypically male. If you don't have it, you are phenotypically female. And don't forget that we're talking about biological sex here. Biological sex, not gender. Gender is a social construct, and it has different ideas and a different basis than biological sex. The Y chromosome, if you can remember from your karyotypes, maybe the X looks like this. The Y looks like a little nubbin. The Y has fewer genes than X. It is smaller. It has fewer genes. But there is one important gene on there called the SRY gene, which stands for the sex determining region. If you have this SRY gene, you are phenotypically male. Because the SRY gene causes gonads to develop as testes. The gonads will develop as testes. The testes produce androgens. Testosterone is an example of one androgen. And then you have male development. You have testes, you have a penis, you have a prostate, you have seminal vesicles. Notice that before all this happens, at 7 weeks, an embryo, you're not quite a fetus yet, an embryo is undetermined. We don't know the sex at 7 weeks. But then at about 8 weeks, this is when the SRY gene turns on, and it turns male development on. If the SRY gene is absent, the gonads are going to develop into ovaries, and that is going to release estrogens, and then you're going to have female anatomical parts. The same thing is true of the external genitalia. With the SRY gene present, at birth, there will be the development of a penis. With the SRY gene absent, at birth, there will be a vaginal opening. Notice that around 10 weeks, it actually looks relatively unclear, similar to 7 weeks. And that's because we all come from undetermined homologous structures where we have similar structures of the penis and the female genitalia. I teach human anatomy typically, and I teach about how males and females have homologous structures. And what that means is that there are parts of the male genitalia and parts of the female genitalia that are similar. For example, the scrotum and the labia. Those are literally the same tissues, except they are expressed in different ways. You know, they come out looking a little different. Another example would be the head of the penis and the clitoris. Those are also homologous structures. They're similar structures, but they have been presented or expressed differently because of this SRY gene. Okay, so let's discuss an issue. We have a normal, healthy girl shown right here. She grows up, she begins puberty, she develops breasts, she develops, you know, a lot of the phenotypically female structures. However, she does not ever start menstruating, and she never really grows much pubic hair. She goes to the doctor to make sure that she's okay because she never got her period. And the doctors do a cheek swab test. And what that means is they're just going to take a Q-tip and they're going to swab the inside of her cheek for cells. And they get cells, you know, they're just going to get some human cells of hers. And what they find is that they have no bar bodies. I know you don't know what that means yet, but we're going to talk about it. She has no bar bodies. So the doctors suggest that she might have complete androgen insensitivity syndrome. Complete androgen insensitivity syndrome. They run further tests on her and it confirms it. She has complete androgen insensitivity syndrome. So let's rewind a little bit and talk about what androgens are. Remember I said that testosterone was an example of one of these androgens. Androgens are hormones, and I know you've heard of the word hormones before, but if you don't know what a hormone is, it's pretty much just a chemical message. It's a chemical messenger. It's kind of like a tweet or something, you know? It's a chemical messenger that gets released, and it's going to signal to other tissues if they receive that signal. And it's going to signal to other tissues to develop male characteristics. So here is an example of what it might look like. Here is our hormone. If a cell is receiving that hormone, it would have the receptor. Only if, you know, it's going to respond to that hormone. It's going to receive the signal. That hormone is going to cause a change where it might produce a different protein. It's going to produce a different protein in the cell, and it's going to make something different. It's going to make the cell act differently somehow. So it's kind of like a tweet, you know, where someone reads the tweet and maybe they get really upset by this tweet. Or maybe this tweet makes them really happy. It's going to change the function of this cell in some way. So when somebody has androgen insensitivity, their body produces the androgens. So they have the testes. Their body produces the testosterone and other androgens. However, their body does not have those receptors in their cells. Their body cannot respond to it. So these individuals have the X and the Y chromosome, but because their body doesn't have these receptors for it, they look female. They are phenotypically female all over. However, they don't act female, right? They don't have a period. They don't menstruate, for example. No menstruation. So they're phenotypically female, even though they have an X and Y chromosome. So this is what androgen insensitivity is. And that image of that female that I showed you before, she has androgen insensitivity. This is a model, I don't know their name, but they have androgen insensitivity. They are a famous model. They've publicly announced that they have androgen insensitivity, which means they have an X and a Y chromosome, but they appear phenotypically female. And I'm calling them a they because I don't know their pronouns. So there is a gradient of androgen insensitivity where it can be complete or it can be somewhere in between. So just to show you what's going on in this individual's body, androgen is being produced by testes. Androgens are being produced. However, there is no receptor. There is no receptor. There's not going to be any change. There's not going to be any new protein made. There is not going to be any response. There is not going to be that phenotypic male traits. So in these, the SRY gene is present, the testes develop, the androgens are produced, but there is no receptor. There is a gradient of this. Complete androgen insensitivity, they look absolutely phenotypically female. And then there's something in between where someone may have partial androgen insensitivity, and this individual may appear more intersex. And so let me show you what that means. Here is a gradient, and it even shows you the gonadal parts to go with it. The complete androgen insensitivity syndrome, there is no response to androgens whatsoever. These individuals appear completely phenotypically female. But if you have any partial response to androgens, and maybe that means some cells do have the receptors, but most don't, and there's a gradient of that. There is going to be a gradient of the expression of male traits. And you can see that with the anatomical regions. And maybe you've heard of, micropenis before. Micropenis can be a phenotypic trait of somebody with partial androgen insensitivity syndrome. Regardless, though, even though these individuals with complete androgen insensitivity look phenotypically female, they don't have the female reproductive system. They are not able to get pregnant. They are infertile, and they have no menstrual cycle. And so they may have breast tissue, they might have little pubic hair, and they might require hormonal treatment depending on the sex that they would phenotypically like to express. Individuals with partial androgen insensitivity syndrome may have male genitalia, but they may be infertile. They may have that micropenis. They may even have female genitalia with pubic hair, but they will be infertile. And so here, the partial androgen insensitivity syndrome, their genitalia is intermediate between male and female. This is the definition of intersex. So this is super relevant to news today. There's a lot of controversy about trans or intersex individuals competing in the Olympics or in other athletics. And there have been things such as sex testing. Sex testing has been done recently to kind of target males that are competing as females. So chromosomally, females should have 2 X chromosomes and no Y chromosomes. This is going to be an extra credit opportunity for you, and I won't say too much of it because I want you to read some articles yourself. But this is super recent. This was in 2018, 2019. It was still a big thing. This individual is Castor Semenya, and I will say that she is chromosomally female, but she is ruled against competing as a athlete in the Olympics. And so I want to ask you for, if you'd like, you are welcome to learn more about her and do an extra credit assignment. There's another athlete here named Maria Patino who had complete androgen insensitivity. Chromosomally, she is male. Chromosomally, she is male with an X and Y chromosome, but she was raised as a female. She actually did not know that she was chromosomally male. Should she be allowed to compete? So, this kind of brings into what we are going to talk about with the second half of this class with ethics, ethical concerns about our genetic knowledge. What are we going to do with situations like this? And who gets to decide? So we have been discussing our chromosomes, our karyotypes, and one thing to point out, that when you miss an autosome, it is always fatal. So whenever you're missing a chromosome 1 to 22, it is always fatal, and the mother is going to have a miscarriage. If you have an extra autosome, it is usually fatal. And the exceptions, if you can remember, are 13, 18, and 21. 13 and 18, though, these babies die at a really young age. And so really, the really rainbow kind of exception here is Down syndrome with the extra chromosome 21. Otherwise, if you have any other extra autosome, it's fatal. These babies do not persist. These fetuses do not persist. But females always have one more X than males. Why isn't this a problem? Why isn't this a problem that we have an extra chromosome than males? This extra X chromosome than males? Well, there is something called dosage compensation. Dosage compensation is when one X chromosome in female XX cells is inactivated. It gets turned off. One X gets turned off. Female cells are only going to have one active X chromosome, and this keeps the X chromosomes in balance. And so it keeps it in the same balance as males. When it gets inactivated, so say we have our X chromosome here, the inactivated one actually gets wound up tight like this, and it is called a bar body. You can see this beneath the microscope. This is the bar body right here. This is the whole cell. The genes on the bar body are not used at all. Bar bodies occur in early embryonic development where a cell is just going to randomly inactivate an X. So that could be the mom's X or that could be the dad's X. and it's going to stay inactive as cells continue to divide. This is at the 32 cell stage, and so this is really early on, right? But then as cells continue to divide, it's going to keep that X inactivated. So it kind of looks like this. There is random X inactivation, and then there's going to be proliferation of those cells. And it's going to kind of make this mosaic structure where different cells have different X's inactivated. So different parts of the body have different genes active just based off of this X chromosome inactivation. So, mosaic cats. Maybe you've seen calico cats. Calico cats are usually XX. They have 2 X chromosomes. I will say this, all calico cats have 2 X chromosomes because coat color on a cat is on the X chromosome. And so what we see here, when we see a calico cat, if you don't know what a calico cat is, it's when they have a bunch of different colors. It's showing you the mosaic expression of the X chromosomes because the coat color is easy to see. So these female cats can express different genes for color on their coats. Male cats only have 1 X, so they're only going to express 1 color. So humans have this mosaicism too, but it is actually not really visual because skin color is not on our X chromosome. One thing that is on our X chromosome is our sweat glands. And so hypohidrotic ectodermal dysplasia can express mosaicism in humans. So for an individual with triplo X, where they have 3 X's, they actually have 2 bar bodies form. 2 bar bodies form. And so that just keeps 1 X chromosome active. Phenotypically female, so individuals with 47 chromosomes with an extra X, they are healthy, normal, phenotypically female individuals. They produce these 2 bar bodies, they have 1 X active, it's no big deal, their body dealt with it, it doesn't, doesn't impact them in any way. So calico cats that have an XXY, they can be mosaic because they can express 2 different colors from their 2 different X's, but they are phenotypically male.